Hello, my name is Tori Kilpatrick, and for my final project, I will be going over how rats are being used in research. My following presentation will be going over what does it mean to have rats be involved in research, what are the pros and cons of having rats in research, and what is the past of having rats in research, what are we currently doing with rats in research, as indicated by a researcher that has recently been doing studies at OSU, and what people in the field expect for using rats in the future. To start off this presentation, I will briefly go over why rats are being used in research at all. So it should be known that rats are commonly used in research so much that 95% of all lab animals are rats is because they are deemed to be the best species to be used as models when studying how pathogens and genetic diseases will affect human populations due to rats being physiologically similar to humans. Some of the pros of using rats in research include that rats have genetic and physiological similarities to humans, as pictured in the genetic tree to the left. They have also helped address neurological and physiological issues, and the studies that they have been involved with have helped us create things such as cancer drugs, HIV, antiretrovirals, and flu vaccines. And they are also a good research model because they are able to reproduce quickly, they're social, they're adaptable, they're omnivores, and they are small in size compared to other animals. So they're able to be stored in very small housing, which is important when you have hundreds per room, which may be dedicated to one study. On the other hand, there are also some cons of rats being used in research, such as even though there is that genetic physiological factor, they're not humans, so sometimes the research that is done on rats will not 100% correlate to the test results that are done with people. Another thing is that millions of rats and mice have to be used each year, which can be taxing to breed altogether. And some people are just uncomfortable with the fact that rats have to be used instead of humans. Now to get into the timeline of how rats have been used in research, we will first talk about some of the past events that have led to rats being used in research. According to the Smithsonian Magazine's, rats have been used in research experiments since the 1850s. Another point is that rat breeders were called rat fanciers because they sold rats to researchers that requested these rats to have certain personalities or unique coat types that would be helpful for the study that the researcher was going through. The Laboratory Animal Welfare Act of 1966 was the first law that was able to protect laboratory animals. Now I will be going over how rats are currently being used in research. And to preface, this section of information was taken from a researcher at OSU's neuroscience department who I have interviewed, who has ran trials with rats with traumatic brain injuries, also known as TBIs, to be able to determine impulsive activity in normal rats versus rats who lack the reasoning portion of the brain. Before any new research was allowed to occur, the researchers involved had to be educated on research papers such as the Rat Park study, which showed that individuals are less likely to partake in addictive behavior when in group enclosures than when they are in an isolated enclosure. And this study showed that when they were in group enclosures, this enhanced the stimuli within the rats. In addition to reading prior knowledge research papers, as mentioned previously, each research staff member must also complete extensive online training courses before even interacting with any of the animals. These training courses include one-on-one rat handling sessions, as well as collaborative institutional training initiative program training, other known as CITI. CITI includes insight into research design and ethics, compliance, safety, and professional development. Next, it should be noted that the principal investigator, other known as API, submits the protocol to the Institutional Review Board, which evaluates human welfare and efficacy from the viewpoint of people that do not have any background in science. This decreases the amount of bias that can happen to allow place for the 3 R's to be used in research, which is replace the use of animals when necessary, reduce the number of animals, and refinement, which is essentially cause no undue harm. OSU has a specific version of the IRB, which is called Buck IRB, and they must approve all aspects of an OSU study before it is able to take place. Now to get into when the research is allowed to actually start. Before the traumatic brain injury, rats are housed together because, as mentioned in the Rat Park study, rats produce the best data when they are in social groups, and they are also identifiable by ear notches. After the traumatic brain injury, the rats are put into single housing to hasten the recovery and to avoid further injury of each other. Rats in this study in particular had weekly weigh-ins, which is important in the beginning when the rats are still adapting to the traumatic brain injury surgery. During surgery, all animals must be under general anesthesia. In this case, it was in the form of the inhalant of isoflurane, and they must also have a secondary form of pain management, such as local anesthetic. In research in general, it should be known that it is a federal requirement for animals in research to be observed and logged daily by qualified personnel, such as veterinarians, to assess the animal's health and well-being. OSU specifically provides 24/7 healthcare by their veterinarians that have been trained with the knowledge of the research protocols put in place for the specific study, which itself gets checked once a month. The rat's bedding and weight are other factors that are checked daily, along with the individual's health and well-being. According to articles in the field, the expectations for the future of rats in research is that rats will be continued to be used by the National Institute on Drug Abuse, NIDA, and the National Institute of Health, NIH, for research on things such as decreasing drug abuse, disease mechanisms, genome editor to repair somatic cells, and many more topics that have not been released to the public at this point in time. Hopefully throughout this presentation you have been able to think about things such as how rats have been used in research, the pros and cons of rats being used in research, and the timeline of how rats have been used in research. Thank you for listening.